Good morning or afternoon, Pape, Daniel, Sebastian, and Anthony will be speaking to you about the cell cycle. Please give them your undivided attention. Thank you and have a splendid day.
Alright, good morning everyone. We're here to teach you about mitosis. Before any phase begins, the cell is performing its regular function in the G0 phase. As you can see here, the cell is producing proteins. The donkey is producing proteins. And then the growth hormone comes in. And the receptor binds to it, beginning the process of the cell cycle. In the G1 phase, the cells are enlarging and the organelles are being duplicated. This process takes about 5 to 6 hours, and you can't see the DNA in the nucleus during this phase.
The synthesis phase. You're currently unable to see what is going on in the cell, so we'll remove it for you. Under the nucleus, the DNA is being replicated and duplicated in order to prepare itself to be separated into a new cell. In G2, the nuclear envelope surrounds and encloses the nucleus. During this, two centrosomes are formed.
This marks the end of interphase. We are now going to get into the different steps of mitosis.
In prophase, the nucleoli disappears. The mitotic spindle is formed, and the sister chromatids come together. As you can see, the mitotic spindle is engulfing the whole cell to make another new identical daughter cell. In prometaphase, the nuclear envelope begins to dissipate and disappears from the nucleus. The centrosomes begin to polymerize more and more microtubules until it reaches the poles. Some microtubules connect to the kinetochore of the chromosomes. These will be later used in anaphase to separate the chromosomes.
The centrosomes are now at completely opposite ends of poles, as seen here. The chromosomes in the center are now aligned perfectly in the middle, in between both poles, forming the metaphase plate, as shown here. In this metaphase plate, each sister chromatid, its kinetochore microtubule, is equidistant to each pole in the cell. Anaphase is the shortest phase of mitosis. To begin anaphase, the sister chromatids split apart. This is the result of the kinetochore microtubules shortening, and each sister chromatid turns into a fully fledged chromosome. These chromosomes are pulled towards each pole and form each equivalent of chromosomes in each side of the pole.
In telophase, the spindles are depolymerized, and the nuclear envelope begins to reform. This marks the end of mitosis, where one parent cell turns into two genetically identical daughter cells. The cleavage furrow has formed. In cytokinesis, the cytoplasm is divided into two. Then the cleavage furrow begins to pinch the cells into two. And now, this marks the completion of two identical daughter cells.
Say you could do better. Tell me, have